Okay, so it is 1 o'clock, and if you're in this panel, this is the Black Mammologist Week speed talk session from three of our Black and Indigenous Scholar awardees. We first have Ilana Mosley. She works on Trypanosoma, Tryp
Trypanosoma.
Trypanosoma in free-tailed Mexican bats, I believe, right? Yes. And then we have Deandra Jones, who works on black bears in the um, Navajo Nation lands. And we're also going to have a recorded video from our Brazilian student, Juan Abreu. He will be talking via video because he couldn't make it to the session, but it's going to be very quick. Just hearing about all the cool, exciting, interesting research that our scholar awardees are doing. And I think we'll just give it another minute to see if anybody else joins. Thank you for joining us, especially on a Saturday. We know that everybody has a lot of cool things to do on the weekend, so it's great that you guys are here to learn about all this really interesting wildlife biology and conservation ecology research that's happening. I think we're going to go ahead and get started at like in 1 minute. So get your snacks, get your water, and get ready to learn. Hello again. Thanks for joining us. Hey, Julian. I think I know you. Um, so again, Black Mammologist Week Scholar Awardee Speed Talk session. We're going to start off first with Ilana Mosley. She's going to give us a talk about disease in free-tailed Mexican bats. So feel free to share your screen.
Okay, can everybody hear me and see my screen all right?
Yes, you're doing great.
Okay, all right. So thank you for the introduction. My talk today will be on utilizing salvage specimens of Mexican free-tailed bats and raccoons for Trypanosoma cruzi surveillance in Texas. Oops. So first to start off with, what exactly is Trypanosoma cruzi? So Trypanosoma cruzi is a protozoan parasite, and it is commonly transmitted by triatomines, most commonly through contact with their feces. And it is the causative agent of Chagas disease, which typically presents as cardiovascular or gastrointestinal disease and can be lethal if untreated. It is what we call kind of a generalist parasite, and it's known to infect almost 180 million species. And it's distributed widely throughout Latin America and the southern US. And it especially has a particular burden in Texas, where it's considered to be an emerging infectious disease. And actually, bats are theorized to be the original hosts of Trypanosoma cruzi. So whenever we think about T. cruzi, other vector-borne and infectious diseases in general, we tend to think about them in three different cycles, where on one end of the spectrum, we have the domestic cycle, and on the other end of the spectrum, we have the sylvatic cycle, and then sort of sandwiched in between, there's the peridomestic. So whenever we think about domestic cycles, it's usually like humans and then animals who are living in very close proximity to humans, so like dogs and cats. And then sylvatic would be like animals that you're usually not going to interact unless like you're going into the woods or like into a certain cave. So like whenever you think about certain cave dwelling bats or even ocelots like what we have here in Texas. And in the peridomestic cycle, they're sort of sandwiched in between. So these are animals that are not necessarily living inside of your house, but maybe are around. So like how Mexican free-tailed bats can commonly roost underneath bridges or even in house attics. And then, of course, our beloved trash pandas, the raccoons. And we're specifically interested in looking into these wildlife reservoirs in the peridomestic cycle because it allows us to better understand and anticipate these spillover and spillback events, which is essentially whenever parasite cycles in one environment transfer over into the next. So first, to start off with, we'll be talking about detection of Trypanosoma cruzi in a population of Mexican free-tailed bats. So back in February 2021, Winter Storm Uri occurred. It was devastating, and it really affected the population of Texas, both humans and wildlife alike. And it caused mass die-offs of a bunch of different wildlife species, but a lot of them were Mexican free-tailed bats. And from that, we collected 283 Mexican free-tailed bats from Central Texas, and we used DNA that we extracted from the heart and also from the kidney to detect Trypanosoma cruzi on a species-specific qPCR assay. Unfortunately, we ended up not detecting Trypanosoma cruzi in this population of bats, which leads us to believe that, at least in this ecosystem, Mexican free-tailed bats are not significantly contributing to maintaining parasite in this environment. One of the most impactful things about this is that, again, these were salvage samples, so we were able to continue learning without the additional need to euthanize any other animals. But we also installed all 283 of these Mexican free-tailed bats into the biodiversity research and teaching collections here at Texas A&M. And hopefully these bats will go on to allow us to learn more about bats in our ecosystem. And if you're interested in learning more about this project, this work has been published, and you can feel free to scan the QR code on our cute little bat friend here. So now moving on to Trypanosoma cruzi in raccoons. So back in 2023, we collected samples from 45 adult raccoons as a part of a hunter-harvested predator hunt. So these were raccoons that were harvested over a span of 24 hours, and then we met the hunters at a common checkpoint, and that's whenever we collected our samples. So we collected heart, clot, and then also a section of the distal descending colon. And from here, we used the same qPCR assay that we used for the bats to detect Trypanosoma cruzi DNA in the heart tissue. And originally, our plan was to have a relatively even distribution of positive raccoons and negative raccoons. So from this, we had 34 that were heart positive, 11 that were heart negative. So we randomly selected 14 raccoons, but unfortunately, our raccoons had other plans. And through further screening of the clot and also the intestinal tissue, we found that we actually only had 5 negative raccoons, which led to a overall infection prevalence of 88.9% in this population, which is like much higher than anything else, like any other peridomestic reservoir that we've observed here in Texas. So now that begs the question of what else can we learn from these salvage samples? And the answer is, in the words of the great Katie Heron, the limit does not exist. So just a couple of examples. So our same bats, one of my colleagues, Dr. Edward Davila, he used samples from these same bats for coronavirus detection. We also have interest in using these bats to learn more about the impact of pesticides on Mexican free-tailed bats. And for our raccoons, we were also able to learn more about Trypanosoma cruzi host-associated strain types. And then we were actually able to do the first characterization of the intestinal microbiota composition of raccoons in North America, and also looking further into associations between different bacterial taxa and Trypanosoma cruzi infection status. And that's pretty much all that I had. Thank you so much, everyone, for listening. I would like to acknowledge, of course, the biodiversity teaching, research and teaching collections, also my sister Iman and my partner Idowu for all of their help with my presentations, and then also various members of the Hamer Lab. And if you have any additional questions, here's my email, and I'll go ahead and take any questions now as well.
Yeah, I have a question. So you mentioned that there's like almost 88%, 89%
Mhm.
prevalence in raccoons. So what does that mean for like human wildlife disease transmission?
Yeah, so it's very significant in terms of human wildlife disease transmission, because these raccoons are playing an important role in maintaining that parasite in the environment. So in the instance of like, whenever you have different, and right now there's not like a Trypanosoma cruzi eradication program or anything like that. But in the instances where you do have eradication programs, it really further complicates things because you have these highly parasitized reservoirs that are keeping that parasite in the environment and also serving as sources of new infections. So whenever kissing bugs are feeding on them, they're kind of like perpetuating this entire cycle and serving as new infections and then going out and then being able to bite humans or dogs and things of that nature.
Thank you. Oh, and there's my alarm. Does anybody else have any quick questions for Ilana?
No
If
questions, but great job. I enjoyed the talk.
Thank you.
if not, we'll move on to our recorded talk from our Brazilian student, Juan Abreu. Just got to figure out how I'm going to share this. I'm going to share this and share the sound. So has everybody seen that?
Mhm.
Okay.
Hi everyone, my name is Juan. I have a master's degree in animal biology and I'm a member of the Master Zoology Laboratory at the Universidade Federal de Pernambuco in Brazil, supervised by Dr. Diego Stu. I was invited to talk a little about my research that involves the mammals known as opossums, which are American marsupials with a small and medium-sized and a wide variation of habitats and habitats and distributed especially in South America. My research focuses on the anatomical description of the masticatory muscles of opossums to better understand the morphological function and evolution of the masticatory apparatus and its relation to feeding ecology of the group. Because opossums have a very similar skull and design, the relationship between morphology and the exploration of the environment requires increasingly detailed morphological studies to understand the evolution of this morphology. So I use the muscle to provide more detailed analysis of the morphology of opossums that is um, much less obvious than specialized taxa such as street carnivores and herbivores. The descriptions of the masticatory muscles are carried out ontogenetically and comparatively across taxa, generating some results that have never seen before. For example, we described for the first time the ontogenetic change in the masticatory muscles at the right ridge opossum Didelphis albiventris from lactaring specimens to chewing specimens and observed the change that corresponded to greater masticatory complexity in the adults as found in masticatory study with the opossums. We also described in the first time the masticatory muscles of a number of opossums, using the muscle to estimate functional factors of the masticatory apparatus as gape, bite force, and mastication via the parameters of the internal muscle architecture and the biomechanics aspects in order to understand how food is obtained and processed for these animals. The anatomy of the opossums we have dissected so far is very similar, but we are observing the individual variations that generate interest in how this variation is reflected in mastigation. This relationship with mastigation is very important because opossums are often referred as having a conserved morphology and maintain a primitive morphology that resembles that of the Neanderthal mammals. So, to understand the masticatory anatomy and the functional morphology of opossums can provide more information about the primitive mastication and how mastication evolved. At this moment, I wanna introduce more complex techniques into anatomical and functional studies, such as 3D visualization and masticatory morphology. This is possible through the use of contrasting agents on the musculature in computed tomography scanning, known as the ICCT, which I'll use in my next project, and I'll make the models available if other researchers want to study. To the Black Indigenous Scholars Award in Mammalogy, I can dedicate myself to study this technique and meet the other researchers who are already doing it in Brazil, including the possibility in visiting them at other universities. So I'm grateful to the Black Mammalogist Week and the American Society of Mammalogists for this incentive for me to continue to study what I like and that should bring more news in the study of mammals. Finally, I would like to thank again the Black Mammalogist Week for inviting me to present here. I would like to thank my incentive in Brazil, and I thank everyone that listening to me. Abraços do Brasil.
Apologies for that, you guys. I don't know what that was. But yeah. So if you guys want to see the video again, we're gonna have this recording up on our YouTube channel. We should have it up by the end of next week. And also, I can send this video, Juan's video, to you guys as well. Personally, just reach out to me. I'm gonna put my email into the chat. Or you can reach out to the Black Mammalogists. Gmail. And so now we're going to move on to Deandra Jones and hear some about black bears.
Great, thank you, Asia. Let me see, 1 second, guys. Okay, hello everyone. My name is Deandra Jones. I'm currently a third-year PhD student. And today I'm going to be talking a little about my black bear project that I'm doing on the Navajo Nation. So just a little background. The project has a focus on the Navajo Nation, which is the largest Native American reservation in the United States, roughly about 27,000 square miles, equivalent to the size of West Virginia. And there's really two main parts of the research, the ecological part and the social part that I call it. For the ecological portion, it will take place in the Chusca Mountains, which is the image on the right side. And the range is about 80 kilometer by 50 kilometer and lies directly on the border between Arizona and New Mexico. The Navajo Nation is in southwestern United States. So in 2011, the black bear was identified as a priority species by the Navajo Nation Department of Fish and Wildlife for a number of reasons. So the first one was that there's no current information about the species and their population on Navajo lands, as the last formal research was done in the 1980s. So we're just really wanting to understand, what that population is today. We want to know whether the population numbers are increasing, decreasing, or stable. So our first objective is really just estimating that population and figuring out how many bears we have in the Chusca Mountains. And there's no management strategy that is currently in place to address these human interactions with problem bears that seems to be perceived to be increasing over the past few years. So just trying to investigate that role of human wildlife conflict to help minimize these conflicts, as well as just, learning about where they're occurring and how often they're occurring within the Navajo Nation. Just we don't have that kind of information. So our second objective is just understanding the human-bear conflict interaction. So finally, participants did note the black bear's cultural importance to the Navajo people. So black bears are considered sacred. Across generations, the black bear has been more than just an animal. It's regarded as a spiritual companion and a symbol of strength and a repository of traditional knowledge, and often referred to as the mountain people. So just trying to preserve these knowledges, as well as how can these traditional knowledge help inform current wildlife management strategies. So our third objective is exploring traditional ecological knowledge, or trying to understand that knowledge surrounding black bears in Navajo culture. So then how are we going to estimate the population? So we're using non-invasive methods only through hair snares or hair traps. So if you guys can see in the bottom left right corner, has that white picture, has trees around it. There's barbed wire. So that's kind of what we're mainly doing. We're setting up barbed wire around trees and hopefully collecting hair. We'll put like bait or lure in the middle to kind of track the bear to either crawl under or crawl over. But with that, you could kind of see the variety of bears that we do have on the nation. So what we'll do is we'll collect their hair, and how we're going to differentiate between these individuals is we'll do DNA extractions from the hair samples, and each of these DNA acts as like a unique marker that allows us for individual identification. And from this, we could do analysis and just kind of figure out how many bears we have within that area. So methods for kind of the social aspect, I call it. So for the TEK, or traditional knowledge, we're going to be doing interviews, just at least 15 individuals, just trying to learn more about the cultural significance of black bears, as well as the significance of these bears to the lands, and any teachings or stories that's related to their role in our creation stories. And then the surveys will be targeted more towards the human-bear conflict, just sending out the survey to everyone throughout the Navajo Nation. This will be paper and online surveys, and just really understanding what conflict is. What people like, is it trouble or is it more of an interaction? Are these interactions good or bad? And just like seeing bears in the wild or living near them, just having that kind of basic information. But just some of the outcomes and impacts, we're preserving Diné traditions or Navajo traditions. We're trying to preserve these knowledge for future generations, just so that they have an understanding of how black bears, are part of our culture, are part of our identity, and just their coexistence with us in the past, as well as engaging with the community, having them involved throughout the study even after, and then also educating them about black bears, creating these brochures, posters, pamphlet explaining our research findings and just the importance of black bears in our culture. We just want to create a better relationship between traditional ecological knowledge and Western conservation practices. But also, we'll have research reports just outlining the findings, and just getting that basic information for the Navajo Nation Department of Fish and Wildlife. But with that, I just want to say thank you and aloha to everybody that has been a part of this journey, that has helped funding, and thank you guys for allowing me to kind of take part in this week. It's been, just really cool to kind of see a lot of the stuff that has been going on throughout the week. But yeah, thanks. And with that, I'll take any questions. Stop sharing.
Does anyone have any questions for Deandra?
I have a question. So the interviews, can you remind me, so is that just Navajo people, or is it also other people in the area of like white people or anyone else?
No, it'll be just for
Okay.
the Navajo community.
All right, gotcha. Just want to remember. And do you find that overall Navajo people have a good relationship with black bears, or do you think like there's influence from like this colonization, for lack of a better word?
Yeah, I see both sides. So I see that there's a lot of people that really understand, these animals, or black bears need to be respected. We need to give them their space. There's a lot of things that we're not supposed to be doing with them. And there's these stories that I hear, and a lot of them are good. They're seen as like I said, like sacred. They're just more of there to protect us kind of thing. But then there's also the other side where, people are like, oh, I seen a black bear, you know, eating my livestock, or, they're damaging my infrastructure kind of thing. And yeah, I think I think it gets really complicated. Definitely if you kind of go deeper into it. But yeah, I definitely see both sides of that. I don't know. I feel like it's hard to kind of explain to them what's happening. Kind of explain, this is sorry. It's yeah, it's complicated, but I see definitely both sides. I hope there's a way that, I could kind of explain it a little better to a lot of the people, saying that, a lot of these problems that we perceive as problems are really not problems. They were kind of from the past, from these issues that we have faced it, and a lot of people like to talk about it. But I hope that answered your question. Sorry.
Yeah, thank you. Thank you.
Unfortunately, my plan is about to cut out. So we have to say goodbye, but thank you again for Ilana, Deandra, and Juan for sharing your cool, really, really interesting research. You did great. And we really appreciate you volunteering for this. So
Thank you.
everybody, again, we should have the recordings up available on our YouTube within the next week. So just look out for that. I hope you guys have a good evening.